outside Earth. This molecule eventually form like amino acid or nucleotide, and then it goes to RNA or DNA or proteins. These biopolymers assemble into protocell. The protocell then slowly evolve into simple cell and then LUCA. Actually, every step is people work on it. We are especially interested in protocells because protocell is, you can see from this picture, before protocells, the molecules are non-living matter. They are studied by chemists. After protocell, they have a simple feature of biology. Therefore, they're studied by biologists. Protocell is a transition from non-living to living matter and from chemistry to biology. That's why it's important. Non-equilibrium. I put a picture here. It shows the currently pretty much all the protocell models, including lipsome, cortisome, palmersome, and so on. When you see them, you know they are not alive because they are at the equilibrium conditions. These two videos shows living things. They are alive because they consume energy. They are at non-equilibrium conditions. If you stop the flush energy, they will die. Therefore, non-living, non-equilibrium protocell holds the key to LUCA. My group is working on non-equilibrium protocells. We take non-living polymers, put them together to form protocell, and then make it to a living matter. To achieve that, there are at least 4 parameters you need to consider. First is materials. You have to choose the material to form the protocell. One shortcut is you just take the polymers from living cells like DNA, protein, polysaccharide, something, and so on. These polymers will automatically form coacervates and lipsomes. We choose coacervate with and without lipsome. The next parameter is mechanisms. You have to choose a spontaneous process for them to assemble into protocell, since there's no hand before life to manipulate those polymers. The spontaneous process including phase separation, self-assembly, and self-organization. We choose currently spontaneous phase separation. The next parameter is living features. There's a lot of them like self-replication, evolution, metabolism, mass transportation, and so on. The key is you have to choose one protocell to have all those living features, or the protocell has capacity to have all of them. The last one is energy. It's energy used to push the protocell from equilibrium to non-equilibrium. The energy we believe has to be physical energy because life has not started yet. But the physical energy includes heating, light, shock wave, and the electric field. What we choose is electric field. Why electric field? Your experiment, the electric field helps to create biomolecules. If you look at literature, you find a lot of statements like this. It says electric fields are found in and surround every living cell, not critical for the proper functioning of biological processes. Those direct current electric fields are present in all developing and regenerating animal tissues. So the electric field are all over our body or the cells, even though we cannot feel them. But they can be detected. I believe everybody take this test once or twice a year. We are happy to see those electric signals. If you have a test that's no signal, I think you are in trouble, right? But we can use even higher electric field to bring back, not always. So electric field is critical for life. It should also be critical for the original life. That's why we choose electric field. For average, you apply electric field. Actually, it's been studied about in 1930s. The drone has reported, in such system, they use gum aromatic and the gelatin. They replace the coagulation and glycalization and circulation. We choose single oligonucleotide and PALL and form coacervate. Thanks for the modern technique. You can clearly see the repeatable cycles of glycalization even fusion. If you go to even higher electric field, you'll see even more fantastic features. Of course, there's also circulation. This is a bilateral circulation, polymer inside circulating. And the bilateral can also transform into 3-zone or 4-zone circulations. With the coacervate and the vacuolization and the circulation, we have achieved several type of dynamic features that have been published, and we also achieved some of them has not been published. I just show them to you. See, it looks like alive, you know. Yeah, moving. Today, one example. Now, it has been published recently in Langmuir. We know all the living cells, even the simple bacteria, they have hierarchical structures. It's not homogeneous. They have membranous organelles. Now, the organelles, they have very important biofunctions. Now, our program, I show you today how we create protocells with membranous organelles. So, this principle. It has been demonstrated that the membrane organelles in living cells are formed by liquid-liquid phase separations. Now you can see it is a hot area in recent years. It happens complex coacervates also formed by liquid-liquid phase separations. They share the same physical principles. If you put different coacervates together, you control the immiscibility. It will automatically form multi-phase droplet. Of course, to tune the immiscibility, you have to, the key is the interface tension and the density. We choose polylysine and the oligo. They have one, they can form coacervates. It's the green one. We choose another polymer. It's polysaccharide. We put a little bit charge called Q-dextrin. It can also form coacervates with oligonucleotide. We use orange color to show it. Dextrin is different from polylysine in two parts. One, the dextrin is more hydrophilic because a lot of OH groups, and we also put lower charge on it, so the charge density is low. Therefore, the droplet, the green one, has lower interface tension and lower molecular density. Therefore, you choose these 3 polymers, just simply mix them together. They will automatically form a droplet with some subcompartments. You can hear the inside is green, is polylysine with oligo. The outside is orange, is oligo with dextrin. And with time, the size increases. The scale by is about 10 micron. You can see 2 small droplets can fuse together with the adjustment inside the subcompartment. So it's because it's liquid. With this system, create different size and the number of subcompartment just tune the ratio of PL as dextrin. As you can, when dextrin is in majority, it's a surrounding phase. The inside the PL and the oligo will have different distribution of subdroplet. Its size and the number are different. They can be different, the same as those in vivo or those in vitro. We can also test the selective sequestration of different molecules. We choose 4 sample molecules. You can see dextrin mainly accumulated in the surrounding phase. The rest 3 is accumulated inside the droplet, a subcompartment. Of course, they have different ratios. Sodium chloride or enzyme outside. The sodium chloride will degrade the surrounding phase because it can screen the electrostatic interaction between DNA and dextran. When the surrounding phase is dissociated, it causes the internal fusion of the subcompartment, as you can see from here. The same thing happens when you put dextran in. It's a degraded dextran, and the surrounding phase has been shrunk, degraded. It pushes the internal subphase to fuse. Electric field can also cause internal fusion, but in different way. As you can from here, at low electric field, it causes a circulation. The circulation push the droplet from the bottom part to fuse, and push the upper part, just push them to fuse. If it's just a many type small droplet, subcompartment, they fuse too, but they have a different shape. If you tune the surface charge of the droplet, you can push the circulation from bottom to the top, because that's the different distribution of the droplet. Sorry, in the subcompartment. Go to high electric field, we can just vacuolization and push the droplet to fission and release daughter droplet either against the electric field, or along the electric field. The released daughter droplets mainly oligo and dextran, because it's released from the surrounding phase. It's not internal phase. You tune the density, tune the surface charge. You can change the number and the size of the released droplet. With this system, droplet with subcompartment, we can achieve internal fusion, fission, some fantastic features. Something missing. We achieve them only in the lab. What are missing is prebiotic conditions, which accounts for the origin of life. We don't know what the prebiotic condition should be. And also we are missing is the local environment, where the droplet should be in the deep sea, thermal vent, or it should go the wet dry cycle, which has been not considered in my talk, in our research. Stop here, and I will use two pictures to show the point of our research. We are working on the protocells. It has to be something live, like a puppy. But most of the cases we are working on something look like a puppy. It's just a toy. For the toy to be alive, you need to reinstall a battery, push it to non-equilibrium so it can jump and bark, look like alive. How to create a living stuff from molecule scale, I think it's a long way to go, but it's doable. This is work done by my students. I also have a lot of collaborators, financial support from the National Science Foundation of China. I'm sorry I cannot take questions now. If you have questions, you can email me. Email me, this is my email. And you are also welcome to my university, so we can have a talk, have a discussion face to face. Thank you for your attention.
All right. Well, that was a wonderful talk. But like we said, that was also a recording. And so if you have any questions, feel free to email Professor Liang. Otherwise, we will go ahead and go on to the next talk. So next up is a talk by Tom Wright, a postdoc from the Shostak group in Massachusetts General Hospital. And he's going to tell us today about stable integration of amino acids into RNA polymers capable of genomic propagation. So Tom, if you're there, please take it away.
Okay, thanks, Albert. How do I do this? Can you
Yep,
see
we
my?
can see.
Okay,
Yep, all
great.
good. Looks good.
My title is a little bit simpler now. So as Albert just mentioned, my name is Tom. I'm a postdoc in the Shostak lab here in Boston. And we are interested in creating life in the laboratory, pretty much as a model system to understand how life could have got started on the early Earth. So no shortage of ambition in our lab. To do this, we work with a model for what we believe could be the simplest system capable of undergoing Darwinian evolution. We call this model system a protocell, and it is made up of two critical components, a fatty acid-based membrane vesicle and a genetic polymer, which we think is most likely to be RNA or something similar to RNA. RNA can both carry genetic information and act as a catalyst for chemical reactions and so-called ribozymes. The vesicle compartment keeps the RNA genetic material localized. If a population of such vesicles exist, one of them may contain an RNA capable of catalyzing a reaction that is useful to the protocell as a whole, shown here as an active ribozyme. This vesicle will be selected by Darwinian processes and will go on to provide the raw material for the next generation of evolution. So the challenge is to make this system replicate. This consists of both replicating the genetic material and the vesicle itself, and also the challenge of coupling the two processes to maintain an integrated unit. I work on the challenge of the copying of the RNA genetic material. There are two ways through which this copying could have occurred on the early Earth, to simplify matters. The first is by primer extension. In this case, a short RNA primer anneals to an RNA template. Extension then occurs through reaction of the 3' hydroxyl of the primer with an sorry, I always stutter on this one, imidazolium bridge dinucleotide, shown here. We use imidazole activated nucleotides as they react orders of magnitude faster than nucleotide triphosphates, and potentially prebiotic pathways for their synthesis and activation are known. The alternative pathway is via ligation. In this case, a short oligo binds the primer template duplex and reacts with the primer. For ligation, in this study, we use methylimidazole activated oligos. Ligation is attractive because it requires fewer chemical steps to copy a given template. However, currently the rate of ligation is much slower than that of primer extension. So the ultimate goal is to have chemistry that can copy RNA templates long enough to encode functional ribozymes, which would enable evolution to have an activity to select for and to get started. There are a number of challenges remaining before we can achieve this. The first is that the rate and yield of the copying chemistry is too low. Within a vesicle, we can only extend up to about 5 nucleotides over about a day of reaction, which just isn't good enough to copy an active ribozyme. The second major problem is magnesium. The copying reaction requires high concentrations of magnesium cations, which acts to deprotonate the 3' hydroxyl and turn it into the reactive alkoxide. However, at the concentration of magnesium ions required to get the copying to work, fatty acid vesicles are degraded. So there's a fundamental incompatibility there that needs to be overcome. We wondered whether amino acids could provide a solution to these problems. Work from the Sutherland laboratory has shown that chemical pathways starting from simple prebiotically plausible feedstocks, such as those shown here, lead to both RNA precursors and to amino acids. Therefore, it is likely that any environment on the early Earth that contained RNA could have had a strong chance of also containing amino acids. Furthermore, amino acids can be covalently linked to RNA via prebiotically plausible reactions of amino acid imidazolides, shown here. With the challenges I have just described in mind, we set out to answer two main questions. First, we wanted to know whether covalently attached amino acids could enhance the rate of copying reactions. It is known from the literature that amino acid linked RNAs can form phosphoamidate bonds by reaction of the amino group with the phosphate of the incoming nucleotide. This is what the structure we form looks like. We thought that this reaction might not require magnesium and could thus be a good candidate chemistry for integration of vesicles. Our second question was whether the products of such reactions, which we call amino acid bridged RNA, could then act as templates for further copying. Such a step is essential if such bridged RNAs are to contribute to continued cycles of genetic replication. So those are the two questions I'm going to talk about today. To answer the first question, we developed an assay to look at the copying chemistry of amino acid conjugated RNA. To do this, we used Flexozyme, a ribozyme that can amino acylate RNA with any amino acid of interest. By treating fluorescently labeled RNA primers with an activated amino acid, in this case glycine, and with Flexozyme, we get a mixture of amino acylated and native RNA. We then subjected this mixture to primer extension reactions and measured the rate of reaction for both the amino acylated and native RNA. As you can see, at 50 millimolar magnesium, the rates are very similar. However, as expected, the rate of the native RNA reaction drops dramatically as you decrease the amount of magnesium ions. In contrast, the amino acylated RNA retains the same high rate of extension, even in the total absence of magnesium ions. This is consistent with reaction taking place via the amino group, which does not require deprotonation by magnesium ion to act as a nucleophile in the reaction. Sorry. This result is exciting as it suggests that it would be straightforward to integrate this type of copying chemistry with fatty acid-based vesicles. We were next interested in ligation. Ligation differs from primer extension in that the imidazolium dinucleotide that binds the primer template duplex and primer extension is replaced by a longer oligo activated with methylimidazole, shown here. For ligation reactions, the reaction with native RNA is so slow that hydrolysis of the activated ligator competes with the reaction, leading to low yields of product. Here, the effect of covalently attached amino acids was even more impressive. In this case, we compared a panel of amino acids. At 2.5 millimolar magnesium ions, the rates for all amino acids tested were 2 to 3 orders of magnitude greater than for native RNA. Intriguingly, for 2 out of 3 amino acids tested, there was a preference for ligation of the L enantiomer, which is the form found in existing biology. Having shown that attached amino acids do in fact enhance the rate of copying, we turn to the second question. We wanted to know whether the amino acid bridged RNA that results from our copying could act as templates for future cycles. We prepared glycine bridge templates using a novel synthetic strategy and tested them in primer extension reactions. We tested 3 cases, shown here, that differed only in the position of the primer relative to the glycine bridge and the template. In the first case, the glycine bridge joins the 2 nucleotides that compose the dimer binding site. So that's sort of the most challenging sort of scenario we could imagine. In the second case, the glycine bridge is located after the primer annealing site. In the third case, the primer is clamped over the glycine bridge. In all 3 cases, we could see copying, as shown by the gel images. These results imply that glycine-bridged RNA can indeed act as a template for RNA copying. We are currently interested in extending these results to other amino acids, which perhaps have more steric hindrance to this kind of reaction. So to summarize our results, covalently attached amino acids can enhance the rate of non-enzymatic RNA copying, especially in the absence of magnesium. The effect is greater for ligation, which is good, as ligation rates are a weak link in the model of non-enzymatic RNA replication. The lack of requirements for magnesium means this chemistry is a good candidate for integration with fatty acid vesicles in our protocell model. Finally, amino acid bridged RNA can act as a template for future copying, suggesting that it could operate as a genetic polymer within a protocell, either alongside RNA or exclusively. So one avenue of future research will be demonstrating this by encapsulating our reaction system within a fatty acid vesicle and showing that the copying activity is unaffected. The more exciting direction, in my opinion, is ribozymes. You'll remember from the very start of my talk that RNA is a strong candidate for the original genetic polymer because it can serve both genetic and catalytic functions. Our ligation results in particular show that attaching amino acids to RNA provides a new solution to copying long sequences that are long enough to form catalytic RNA. We have shown that non-enzymatic ligation with different amino acids can furnish RNA strands with a range of side chains. This may provide new opportunities for ribozyme catalysis to make use of functional groups that are not available to RNA, and thus expand the chemistry available to early cells. So I'd like to thank you all for the attention. I've tried to keep it brief and punchy. Especially like to thank Jack, obviously, for taking me in and being a great mentor. All the work presented here was done in collaboration with Alexander Radokovic and Victor Lilabel, and I'd be really happy to take any questions. So do I stop sharing now?
You don't have to just yet if you want to show some slides for questions. So we do have times for questions. And first of all, thank you, Tom, for that wonderful talk. So I can start off with a couple of questions.
Okay.
So I was always wondering about the triphosphates, and I know that they're slow, but when we think about deep time, and even not so deep time, just on the order of years or something, is it really, are the triphosphates really not going to work? I mean, I know Jack published a couple of papers on this in the 90s, but even then the ligation seemed to work. It was just slow. So I was wondering if you could comment on that anymore.
Well, with ligation, that's a good point. I think it doesn't matter the time scale, unless you're getting on the orders where RNA is hydrolyzing itself. I'm not sure of the exact rates. The problem the other problem with triphosphates for primary extension is that they don't really get into fatty acid vesicles, as I understand it, because of the charge.
Okay.
They're less permeable.
So you have to use the imidazolides.
Yeah, they can cross the membranes, as far as I understand. I'm not really a vesicle guy, but that's what I understand the argument is.
Cool. And my other question, if nobody else has any questions. Oh wait, there's one from Anne. Anna, let me ask. So she says, Tom, amazing results. Do you know if the amino acid bridges can affect activity of ribozymes, e.g. secondary structure?
No, so that's something we're really interested in looking at. The problem we have is synthetically accessing ribozymes with these amino acid bridges is still quite difficult for various technical reasons. But yeah, it would be really great. There's been examples of people doing in vitro selection with non-covalent, just putting amino acids in the solution and showing that amino acids can be recruited as cofactors by ribozymes. The other difficulty in doing it the way we'd do it is we'd start with like a ribozyme sequence. So you'd be just chucking in amino acids kind of by design, which is never the most efficient way to get things done. Really, we'd want a way to be able to evolve ribozymes that have amino acids. And people have done this through like chemical modification strategies, but those are obviously not prebiotic chemistry. So yeah, we're currently pursuing, we have some interesting results. We've shown that you can assemble a ribozyme through a series of ligation steps using our amino acid bridges, and that the ribozyme is active in its native activity. So they're tolerated, but then extending it to use the amino acid as chemistry is kind of a technical challenge that we'd really like any good ideas on.
All right, thank you. We have time for one more quick question, if you can answer it quickly. Yeah. Harold Bernard asks, what pH are you doing these amino acids containing RNAs at?
So all the reactions I've shown are at pH 8, which is just standard for primary extension and ligation.
Okay, cool. And there's a couple more questions in the chat that you might want to look at, but we should start moving on. So thank you very much again, Tom. That was great.
Thank you.
So next up, we have our own Anna Wang from the University of New South Wales. And she's going to tell us today about building protocell communities. So Anna, please take it away.
Hey, thanks, Albert. Yeah, I'm super excited to be presenting here. Kind of still stoked that this conference has happened. So thanks everyone for participating. Yeah, I wanted to talk about like a pretty sort of coronavirus-y project, and that's me being super honest. Like, I'm not sure I would have looked into this more if I weren't, stuck at home. But yeah, so this is kind of born out of just staring at some pictures. So the previous two speakers have both spoken about protocells and cells and given really great introductions there. And basically the reason why we want to focus on this sort of structure is because cells are the fundamental unit of life. These unicellular organisms do wonderful fermentations and make food delicious for us. And we are multicellular organisms, so examples of cells cooperating together in a way that we don't quite understand yet, and making these, individual entities that are the sum of all of these cells interacting in concert. So if we look at this fundamental unit, so Tom had some really great schematics before. The essentials are there's some nucleic acid inside like a bag, and that bag kind of forms the boundary and delineates what this entity is. So this is a cartoon that's often found in biology textbooks, and you'll see that the cell membrane or the plasma membrane is often shown as a sac. And what it consists of at the molecular level is a phospholipid bilayer. That means there are two leaflets, or like two layers of these amphiphilic molecules, and they're quite thin, so you can barely see them under, pretty specialized types of microscopy. And if we were to blow up this cell to the size of a house, so this is actually the house that Tony Jaa from LC lived in when he was a graduate student. It's a beautiful Boston house. So if you blow up a cell to the size of a house, the cell wall is pretty much as thick as an iPhone. So these are really, really thin structures, but they're pretty tough. If we compare it to the mechanics or the physics of these large scale structures such as houses, cell membranes are really quite remarkable because they form that boundary. They keep the insides in, they keep the outside out, but they also do a lot of active processes like transport nutrients inside, like my student Lauren Lowe was talking about yesterday. They also contain organelles that help move cells around, like my other student Daniel Liu was talking about yesterday. And the cell membrane does all these different processes. It's super thin, it houses a great number of proteins that perform all these functions that are essential for life as we know it. So although it's important for a cell membrane to sort of delineate that individual, and because it's important for these sort of individuals to have a sense of identity, it's also important for communities to be built amongst cells. And Bruce Damer touched on this yesterday during his great presentation about origins of life in hot springs. So you get a lot of advantages when you have cells in communities. It's good for them to retain their own genetic identities and selective advantages. But say there's one sort of weird protocell or cell that has a particularly special chemical inside that it's generating, or some other selective advantage, it can be shared if everyone else is closer by. Otherwise, it just gets diluted into solution. So if it's dilute and this cell bursts or it's just a bit leaky, then those molecules are kind of lost forever. So that's just one reason why having communities of cells might be important. And so the question that I wanted to sort of discuss today was, how do we start building a community of protocells? Just for context, my background is physics, so I literally think of things as circles and spheres. And the thing that jumps out to me about lipid vesicles, or these bilayer lipid membranes that surround our cells, is that these structures are what we call colloidally stable. If you have a bunch of oil droplets suspended inside water, or water droplets suspended inside oil, and you leave it for long enough, those droplets will coalesce into a separate phase. And the thing about cells is if you put two of them next to each other, they might get close to each other, but they're not going to just spontaneously merge. They keep that sense of identity, and that's something quite special about lipid membrane-based compartmentalization. And so in order to start building a cell community, there must be some sorts of attraction or adhesion between these membranes. So this again is sort of the physicist picture of what adhesion is. So when we have cells in water, or any microscopic objects in water, they undergo Brownian motion, which means that they're constantly jiggling. And if there's no adhesive interaction between two objects, they'll just keep jiggling. They'll maybe encounter each other and then they'll just move away again and explore space through Brownian motion. If they experience strong adhesion, then when two of them meet each other, they'll just stick irreversibly. what happens then is this whole structure will undergo Brownian motion, and it'll keep on mopping up objects as it explores space. So if a sphere, if a cell hits that aggregate, then it'll just get stuck and it'll start forming this fractal shape. And that's this fractal sort of aggregation is quite common when there's a strong adhesion there. And there's this sort of Goldilocks zone in between where you have weak adhesion. So, you know, these cells, they're kind of attracted to each other, but not really. So the jiggling still gives them their individual sense of freedom and they can explore space, but not too much. They're kind of still held on by their neighbors. And there's a way to quantify this sort of Goldilocks zone. When I say things are jiggling around, that's because of thermal fluctuations. So things will jiggle around more when it's hot and it'll jiggle around less when it's cooler. And the way we quantify this numerically is by multiplying the temperature by the Boltzmann constant. So there's this quantity which we call KT, and it gives you the idea of how strong thermal fluctuations are. So if the adhesion between two cells is weaker than the thermal energy KT, then nothing sticks because the jiggles win, the thermal fluctuations win. If the adhesion is much stronger than KT, then thermal fluctuations don't really matter. If two things collide, they will stick to each other. And this Goldilocks zone is when the adhesion strength is about order KT. And that means these rearrangements are possible. So the cells are loosely stuck to each other, but thermal energy will allow it to slightly detach, maybe move around a little bit, maybe translate or roll a little bit. And what that means is that this system that exhibits weak adhesion can find its ground state. And that's basically saying it's able to find its lowest energy state. And in the context of making protocell communities, what this means is if there's weak adhesion, then you can maximize the number of contacts between cells. So you can see to the picture in the picture in the middle that for the strong adhesion case, because the adhesion is so strong, the cells can't roll around and explore space. And each one of them is maybe only connected to 2 or 3 other neighboring cells. Whereas for the weak adhesion case, it can range even like a crystal and have many, many nearest neighbors. So for building a community, you know, we want a weak source of adhesion. So to explore this experimentally, we use a fatty acid model system. So again, Tom mentioned that we, in the Origins of Life community, are big fans of using fatty acids to make bilayer membranes, and many talks yesterday also touched on this. So this is a microscope image of some fatty acid. We can call them protocells, I guess. So there's green fluorescent dye labeled RNA trapped inside a red membrane. And these membranes are really thin, like 3 nanometers. And at the molecular level, it's again a bilayer, just like a phospholipid. And for those who haven't worked with fatty acids before, I bet you actually have. If you've used a bar of soap, then you know what fatty acids are. So a fatty acid, if you increase the pH, then it becomes negatively charged. And when that negatively charged molecule interacts with a cation like sodium or potassium, then we call that a soap. So fatty acid, soap, depends on what pH you're working at. And for those who like chemical structures, what I show here is the structure of POPC, which is a phospholipid. And if you chop off one of its tails, it's actually a fatty acid. So this is a molecule that is, you know, found within us. We eat them. They make up our cell membranes and the triglycerides. So what I'd just like to show you quickly is we can sort of calculate the interaction energy between fatty acid vesicles. These things are actually very negatively charged. So they're pretty stable. You can see in this picture here, they're kind of near each other. They're not just like melting into each other. But basically, if you calculate the interaction potential between 2 negatively charged objects, they will repel. And what this red line means is as you bring 2 fatty acid vesicles closer to each other, they'll experience this huge amount of repulsion. So that line goes way up, and it means it takes a lot of energy to bring them closer together. If instead we consider, you know, there are lots of small vesicles around, there's this sort of entropic interaction called the depletion interaction that can happen if you have lots of small objects near a big object. And what this depletion interaction does is it actually makes it energetically favorable to have these 2 vesicles come closer together. So in that black line, you'll see 2 vesicles that are moving close to each other. And then they'll experience an attractive well. So that dip below 0 means that they experience an attraction. And that's quite nice, except you'll see that the attractive potential there is 1% of 1 kT. So that's basically nothing. Thermal energy is going to win. So the thing about these fatty acid membranes is they can actually grow in response to stress, which, you know, if most humans get stressed, they like eating. So we also grow in response to stress. But anyway, totally irrelevant. If you have a fatty acid membrane, it can actually stretch. And as it stretches, it'll incorporate more lipids and it can actually flatten out. So if for some reason there's some weak attraction between two vesicles, they can actually start flattening out against each other. So what I then calculated was the interaction between two vesicles that are flattening out against each other. And basically in the beginning, you just get repulsion if you don't consider depletion. If you do consider depletion, then you actually see that there's this attractive potential well, which means that as the vesicles flatten out more, it's like energetically favorable. And if we calculate the energies for typical vesicles in my samples, you'll see that if we allow vesicles to flatten out, then the interaction energy or the adhesive energy can be about 1/5 of kT, which is getting us closer. So I'll show you what happens if I scroll through a sample of vesicles. And yeah, students in my lab have also sort of witnessed this. So it's a very, very faint video because I've had to process these images in order to see the membrane. But you'll see when we get close to the top, suddenly we see something that looks like a tissue. If anyone's looked at onion cells under the microscope, they kind of look like this. It's a huge network of vesicles. And if you look at the image on the top left, you can see these as fluffy, cloudy aggregates in our samples. And so basically, although the interaction energy between two adjacent vesicles is very weak, it's a fraction of a kT. Thermal fluctuations win out most of the time. If you start forming networks with these, if you start giving one of these vesicle membranes many partners to adhere to, then we can start giving them lots of interactions, and that total interaction energy then beats kT, and you can start having these networks form. So if you want to add one vesicle to one vesicle, they're not going to stick. But if you add one vesicle to a network, then it will stick. So the take-home message basically is we can get these model primitive cell membranes to form networks. The main reason is because they can actually grow. And the second reason they can form networks is they don't really want to stick to each other. They're only kind of attracted to each other, but if you have lots of them together, then it works. So I'd like to take any questions, and thank you all very much. And just to give a shout out to my students who also do really great work. We're trying to basically create this whole life cycle out of soap bags, essentially. So yeah, thanks, Albert.
Thank you, Anna. Wonderful talk. We have time for maybe one quick question. I can certainly ask one. How, or do you think it's possible that we can start putting reactions inside of these vesicle networks and see what happens? You know, I'm thinking of like something along the lines of some beta keto acids or some keto acid structures reminiscent of citric acid cycle or something like that.
Yeah, let's do it.
Okay.
I'm serious. Like I might be your colleague in chemistry, but I don't know nearly enough. If it's compatible with fatty acids and they just don't implode, then yeah.
Yeah, I think so. I hope so. Anyway, well, thanks again, Anna. I guess we should move on. So our next speaker, or pair of speakers, I guess, are Rosa Reyes, hope I'm saying that name right, and Diego Orihuela. And they are from another continent, South America, from Colombia and Peru, I presume, respectively. And they're going to tell us today about the Cenancestral population concept, discussing LUCA's ambiguities. So if you guys are there, go ahead and say hello.
Hi.
Yes.
Hello.
Hello, Albert. How are you?
So, pretty good. Hope you guys are doing well. Feel free to go ahead and share your screen and we can get started.
Okay.
So before we start, well, hello, my name is Rosa. So if you all please can go to the link I just put in the chat, so you can submit an answer of a word that you relate with the concept of LUCA. So can you please go do that now? And let me know maybe if you have any problems. So yeah, let's start. Thank you very much.
Okay. Hello, everyone. My name is Diego Orihuela. I'm a member of the Peruvian Scientific Society of Astrobiology. And the title of our talk is the Cenancestral Population Concept Discussing LUCA's Ambiguities.
Yeah, so I'm from the Planetary and Astrobiology Science Group for the National University of my country, Colombia. So yeah, let's continue. So I don't know if any of you were able to submit an award that relate with the concept of LUCA. Can you please let me know? Yeah, okay. So Diego, I don't know if we can show the result. Okay. Yeah, thank you very much. Yeah, of course, Lauren. We okay. So maybe we shall continue. At the end, we show the answers. Diego.
¿Estás viendo la pantalla?
Yeah.
Okay.
So well, I'll just start right ahead while our slides get in order. So when asking about the attributes of life of the common ancestor, we often make like a confusion between the ancestor and the ancestry. So we just assume that we have ancestry, a shared ancestry by when studying the common ancestor. But what can be said about the distinction between these two questions, and how is the concept of LUCA helping us to deal with these problems? So by defining a precise distinction, we can enrich our inquiry methodology, and this is really important for a science as astrobiology. So here today we are just on early career scientists that bring this question to you. So maybe you can help us set the common grounds for addressing this subject. So what about, well, I believe our slides have a slight issue, but okay, excellent. So we want to define how to ask and what to ask, because we believe this will help us into reconstructing this historical explanation. I have you have seen this graphic before. It's from an awesome article that is called Life with for things to take into account. So continue, please.
Okay, understanding the ontology of universal common ancestor. To understand the ontology of the universal common ancestor, referring to its attributes and characteristics, it's important to determine the location of the root on the tree, where LUCA would be located. A number of more recent studies have strongly indicated that the eukaryotes arose from within the archaea. Probably as the result of some kind of fusion of an archaeon with a bacterium. This leads to a two-domain model of the evolution of life, which helps to solve the problem of the order of the branching between domains. But still leaves open the question, what does the universal common ancestor look like? To answer this question, here we show three possible models for universal common ancestors. The genome of Eden is a single genomic ancestor of all extant life. This would clearly count as LUCA. However, it's not empirically reasonable because it's unlikely the existence of a single cell that contained ancestral copies of every gene in existence today. Despite this, there is still the idea of a LUCA with these characteristics. On the other hand, cellular LUCA is a single cellular ancestor here, but it's not a genomic LUCA. Other cells in other lineages existed as well and contributed genes to LUCA's lineage via horizontal gene transfer, represented here by horizontal arrows. These other lineages have since gone extinct, leaving only descendants of LUCA. The progenote term here marks the idea that the genotype-phenotype link had yet to complete its evolution. The usage of progenote as simply another name for LUCA is incorrect. Organismal and molecular common ancestors are consequences of the pattern of evolutionary history and methods used to analyze it. The presence of HGT, or horizontal gene transfer, makes difficult to trace organismal phylogeny, and using single genes as phylogenetic markers is insufficient to reconstruct evolutionary history. The molecular phylogeny with horizontal gene transfer, in blue here, is different from the Cenancestor of the organismal lineage in red. And the time at which these last common ancestors exist also differs. HGT has a key role in these differences. Saksibajeva et al. Conclude, for example, that each molecule, nucleic acids and proteins, has its own history and traces back to an individual molecular ancestor.
Okay, so an example of how to deal and how to cope with these organismals ancestor, where we dive into the three main branches of canonical branches of astrobiology for defining life's origin. So metabolism first is one of those. And here I show one of the amazing examples among this branch of information that is not passed by phylogenetics, that we cannot trace back with phylogenetic tools. So they propose this microspaces unit that is a physicochemical unit that will act and start to make emergence of properties related and typical associated with life. So we just wanted to ask, how can we study the differences of these phenomena? So continue.
Here, more ideas about the progenote. The progenote proposed by Woos and Fox is a group of cellular or procellular entities with rudimentary traduction systems susceptible to errors, which would have generated diversity. Genotypes and phenotypes were not linked, and progenotes could share structures like proteins due to fluidity of membranes. A progenote wouldn't be an individual in the biological sense. We can think of an organismal LUCA model, assuming that LUCA was a cell whose ancestors had already crossed the Darwinian threshold, while the Woos community-style LUCA model asserts that LUCA was a community of precellular and pre-organismal progenotes. This cell and pre-cell distinction marks an important difference in views about LUCA. And here, two ideas, one of Woos, who said the universal ancestor is not an entity, not a thing. It's a process characteristic of a particular evolutionary stage. And another of Doolittle, who said, I too think that LUCA was a population, but I argue that this in fact means that there actually was no LUCA.
Okay, so well, what is the astrobiological relevance of all of this? Well, we might be thinking that from our scientific assessment that knowing the ancestor nature will allow us to interfere its possible configuration. Oh, we run out of time. So just wanted to show quickly that, please continue, that we have more or less similar representation of this phenomena. This is actually a graph that I show here by Dita. I hope that you can tell me the author because I know I saw it before, so continue, and it's amazing.
Okay, here final remarks. One, the ontological knowledge of the Cenancestral, referring to its biological attributes and characteristics, sorry, is important to facilitate its conceptualization and establish consensus for its definition. Two, the variety of terms used referring to the universal common ancestor of all living beings could cause distortions that make the flow and development of its knowledge difficult. And three, as a community, the precise definition of the subjects and context of astrobiological research is important for dealing with the self-referencing nature of life's studies as it is known on Earth.
Yeah, so please make questions. And thank you very much for having us. It's really a great honor to be here with authors of papers we read back here in Latin America.
Thank you so much, all.
All right. Thank you both for that wonderful talk. And we have a couple of minutes for questions. So one question I have is, if the LUCA was not an entity or but a population or a process going on, is it still possible to try and pinpoint where or what geochemical context it actually evolved from, or is it too late at that point? It had already spread to many corners of the globe.
That's a great question. I'm just trying to deal with the thing about the hot springs subject and how if I mean for me it's really interesting, really important the energy side of life, and if without the configuration of this proton gradient, how can life spread airways where it does not have these conditions when it's like in this, I assume, fragile state, initial state. So this dependence of the environment. So it's just a subject that will take me a lifetime to answer, perhaps.
Yeah, it's very, very interesting. All right, well, if there's no more questions, let's go ahead and move on so we can stay on time. So thanks again, guys.
Thank you very much.
Thank you.
It's great to have you. So next up, we have Fouad El Baidouri. I'm sure I butchered that pronunciation.
No, it's
sorry.
Perfect.
Oh, I got it. Okay, good.
Yeah.
So Fouad is he's coming from the University of Hawaii today, virtually that is, and he's gonna talk about the phenotype of the last universal common ancestor and the evolution of complexity. So please go ahead and take it away.
Thank you. Can you see me at the same time, camera, because I couldn't see myself? Okay.
I can see you right now, but I can't see your slides.
Oh, I should put a slide share. Sorry, it's on Zoom. Share screen. Yep. Okay.
Okay, now I can see your slides, and yep, all good.
Perfect. Thank you very much. I'm really happy because this is my first astrobiology conference. I was dreaming about that for a long time. So I'm glad to be here. And thanks for Rosa and Diego, because I will skip some slides. I'm Fouad El Baidouri. I'm postdoc at the University of Hawaii. I work in AAM lab, and I work on marine yeast that we find also in deep thermal vents and in Antarctica and in different environments. These yeast are the agent of malassezia, have dandruff, and we call it malassezia. So there is some things that I will just mention briefly that Rosa and Diego talked about. So there is this vision of 3 domains of life, and there is this concept of Darwinian threshold. Although it's controversial, I use it just to tell that today I'm talking about LUCA, the last universal common ancestor, or the most common ancestor of all living organisms today. And prior to that LUCA, we can talk about progenote, we can talk about the origins of life, and RNA, DNA, hybrid world, whatever we want. So I'm talking about the Cenancestor, or the most common ancestor to all living organisms. All of you guys have seen this picture of 3 domains of life by Was It All. And the discovery of Asgard Archaea a couple of years ago, or a little bit more, has changed our view. And we moved from this 3 domain of life to the new model that Fraser yesterday gave a nice talk about it, which is like you have 2 primary domains of life, bacteria and archaea, and eukaryotes being a secondary innovation in terms of evolution. So there's different models about eukaryogenesis, and Fraser gives an alternative hypothesis yesterday. Today, I will focus about that common ancestor that link bacteria and archaea with the last bacterial common ancestor and LUCA, the last archaeal common ancestor. This is the 3 ancestors I will talk about today. And in order to reconstruct to go back in time and reconstruct what looks like LUCA, there isn't many approaches. So either if you are a paleontologist, you will use fossil records, but unfortunately we can't use that for microbes in order to reconstruct the past. So this possibility being not helpful, there is the alternative possibility, which is using genomes and gene composition in order to reconstruct the last universal common ancestor. When I'm talking about the last universal common ancestor, obviously I'm talking about a population of cell. I'm using like a metaphor to say like as an individual, but we're talking about a population of cell, and I will say the population of cell that at some point have been very successful and take over the rest of the possible life if it existed. So recently, or in 2016, Was It All in the William Martin's group have reconstructed the genome of the last universal common ancestor based on the fact that it's the common ancestor of only bacteria and archaea. So they reconstructed a minimal primitive cell. And I have to tell you that as a biologist, the pervasive perception of LUCA as simple cell is anchored in our psyche. So we think that this is very simple, and I understand that because an evolutionary Darwinian process should continue gradually from simple to complex forms, which is what happened in most of eukaryotic life. So this common ancestor that Was reconstructed was anaerobic, CO2 fixin, nitrogen fixin, and was maybe thermophile, although David Deemer had wrote a letter about it called Was LUCA a thermophilic progenote? And it was issue dependent, and it has all the other characteristics, cumulotroph, and living probably in hydrothermal settings. So this is the first really attempt using genomic approach in order to construct what looked like LUCA, its physiology and habitat. Prior to that, there was some speculation, there was some attempt using gene absent presence, but this is the real first attempt to present a realistic approach for LUCA. However, the presence and absence of genes alone cannot tell you about the phenotype of the last universal common ancestor. So in our case, what we did is we tried, instead of using genomic approach, we used phenotypic data and tried to reconstruct what LUCA looked like based on phenotypes. So in order to do that, we used what we call the ancestral state reconstructions. So basically the approach is you use observed traits for extant taxa of bacteria and archaea using different evolutionary models and a phylogenetic tree with branch lengths, and you try to infer probabilities about the ancestral states. We did this also in collaboration with my colleagues in University of Reading. So this approach has been validated in different organisms. It was used for diatoms, for example, and using parsimony, or used to reconstruct the morphology of hermit crabs, or even we used it recently to reconstruct what looks like the ancestor of Firmicute bacteria. So while this approach was mostly used for eukaryote, there is these two examples where it has been used for microbes. Prokaryotes. So to reconstruct this phenotype, we collected data for over 3,000 bacterial and archaeal species for 22 phenotypic fundamental trees. I went through all the volumes of Bergay's manual systematic bacteriology and also the LPSAN database. So we collected data like, for example, shape, motility, habitat, oxygen requirement, in ACL, pH, a lot of different characters, both categorical and continuous, plus genome size, gene size, and protein size. So using this ancestral state reconstruction, and for that we use what we call the phylogenetic comparative method in a Bayesian framework. So we attempted to infer what looked like the last common ancestor using phenotypic approach. So our reconstruction suggested that LUCA indeed lived in aquatic environments, probably in hot environments. We suggest it's a hyperthermophile, although it's still there is some debate over this. It was halotolerant with the salinity less than the current salinity level. It has a neutral pH. Like LUCA, the last common ancestor of and it was anaerobic, chemolithotrophic, and surprisingly, it has catalase and oxidase. Although the concentration of oxygen at the time of LUCA is thought to be like 10 to minus 3, the actual levels of oxygens we have today. This is some of the estimates. So the last universal, the last common ancestor of archaea has also similar characteristic to LUCA in being halotolerant, hyperthermophile, not living in neutral pH. It was not clear whether it was anaerobic or aerobic, but it was also chemolithotrophic and has catalase oxidase. The last bacterial common ancestor has also similar characteristic to both LUCA and LUCA, but was not hyperthermophile, was likely thermophile. And when I say thermophile, it's about definition. It's between 40 to 51 degrees Celsius. It was also anaerobic and chemolithotrophic and has oxidase, which is also surprising here. So now, how did it look like? So this is about physiology, about habitat, what about its actual shape? How did it look like? Because all what we know about LUCA is speculation. So as you know, the diversity of bacterial archaeal shape is impressive. So there is a huge diversity, but we can reduce these categories to 3 main categories. That is to say, coccoid, the true spheres that can just divide, or rods that can elongate and divide, or ovoid that can elongate a little bit, have a septal growth, and then divide, like its intermediate structure. So our reconstruction suggests that LUCA was probably an ovoid and motile cell. It has a cell wall, and it has a monodermic cell plan. That is to say, it has single membrane, but it was not clear whether it was spore forming or not, though it was spore formation is considered as an ancestral state. So it was small in size, and it has about 2.5 megabase genome of 2.5 megabase pair. So the last archaeal ancestor has also similar genome size as LUCA, was ovoid motile, has cell wall also, and has a single cell membrane. However, the bacterial common ancestor, as it was suggested before, was a rod-like motile and has a genome that is slightly bigger than that for LUCA and LUCA. But it was not clear whether it has single membrane or not, and was non-spore forming. So my conclusion is that given the Earth formation timeline, given the estimate of LUCA and some of the earliest fossil we have, if life appeared on Earth, it must originated in Earth, it must have evolved very quickly, considerable cellular complexity in less than 500 million years. That's the main important point. So even if some people have suggested that LUCA might have been complex, it's about speculation. Here we provide for the first time a different approach based on phenotype and also using a different method to suggest that LUCA is actually probably a complex cell, as complex as many modern bacteria and archaea. Finally, the challenges of being small. This is a very important point based on the fact that I worked in a lab of biophysics for 5 years, and I've been in touch with the people like some of them are known, like Roman Stoker or Justin Seymour or Martin Ackerman, people who works on microbes and physics. So they combine both. So even if Dobzhansky say nothing in biology makes sense except in the light of evolution, I like to say that nothing in evolutionary biology makes sense except in the light of physics, and I tell you why. Because when you come to the physics of microbes, that is the world that they experience, it is completely counterintuitive from what we live. For instance, if gravity is a fundamental force during evolution of macroscopic organisms, plants, animal, whatever we want, in shaping muscles, skeletons, for microbes, it's negligible. Gravity is not a fundamental force in their evolution to my perception. So they live in, for example, a highly viscous world. If you are a fish and you swim back and forth with your fin, you will move. But if you are a bacterium and you do the same movement, you will stay in your own place. As Anna said, there is Brownian motion. They are constantly bombarded with particles and they are moving. There is molecular diffusion that will limit their nutrient uptakes and access to nutrients. There is also kinematic reversibility, as I said, so where your movements cannot be like back and forth. You have to do like a screw-like movement with a flagellum or something like that. So all of that is to say that the physics of microbes is fundamental. And I think even it is important that in astrobiology or in microbiology class, there must be some sessions about physics of the microscale because it's completely counterintuitive and implies different evolutionary force acting on microbes. Finally, if we look at microbes in space, as we will see in the next session, I will expect that it will be microbial. And if it's similar to life form, it will be similar to bacterial or archaeal species, only if it's similar to life on Earth. Finally, I would like to thank my colleagues and my previous supervisor, Stuart Humphreys. And a last thing, I would like to introduce you to the new school .space, which is a nice group of people from the new school in New York who works about explore the dynamics, distribution, and egalitarian models of governance and design on all aspects of space exploration. Thank you.
All right, thank you very much, Fouad, for that excellent talk. And I have a question, and I think there's a couple questions in chat, but for the interest of time, I believe we should go ahead and move on. But thank you again.
Thank you.
So next up, we have Bryan Drake from the University of Auckland. And he's going to talk today about, or a talk titled On Acknowledging the Fourth Domain of Life. So Bryan, are you there?
Yes, I am, and my screen should be up. Can you see that?
Oh yeah, perfect. We're all good.
Fine.
Please take it away.
Okay, I'm Bryan Drake. I'm a research associate working with Kathy Campbell in her Hot Springs research group at Auckland University. And today I want to propose a change in the way we classify all living things. Now, I thought I had a novel suggestion, but Aditya on Monday had the same classification, and I've seen it crop up again this morning, so it's not all that novel. But nevertheless, let's go through and have a look at it. Now, here we are. We've got 3 domains of life. That's the way it's normally categorized. And what I want to do is propose, hello, don't want that, sorry. What I want to do is propose that we go to a 4th domain with vira, viroids and prions. Now, just to make sure what we got here, a virus is a protein shell, a viroid is a small segment of naked RNA, and a prion is a small protein segment. And the first thing we need to think about in this is a virus living? Are these things actually alive? Again, we'll follow on that, looking at whether life is a rare chance event, what was LUCA, and what drives evolution. So let's begin with this question of whether a virus and these prions and things are alive or not. And you can argue about this for months. That's not worth arguing about because in actual fact, it's a semantic issue, not a scientific issue. Sheldrake, in one of his books, says that many scientific concepts lack a stable definitions, but remain helpful categories to think with. And in that, he puts time, chemical bonds, genes, and species. All of these lack stable definitions, but remain categories which are useful to use. Now, viroids, and prions all share complex chemistry with living things. So amending our tree of life by adding a 4th domain would acknowledge this connection and force us to more seriously investigate the place of vira in the origins of life. So is life rare or is it likely to crop up more frequently? Well, Jacques Monod back in 1971 was quite clear on this. He said the biosphere is a product of a unique event. He said the universe is not pregnant with life. He thought it was a very, very strange thing that life had happened and was unlikely to ever occur again. And he is wrong. I proclaim that life is a thermodynamic necessity. The laws of physics require it. The second law of thermodynamics describes the fundamental trend of the universe. It's that from order to disorder. It's an inevitable progress to increased entropy. So how do we get life? Well, we get it because we have energy put in to do work. Work creates a localized order, but always the effect of work is that there is an increase in entropy in the wider environment. So when you make life, you are using energy to create a greater order in a particular situation, but the effect of that is it increases the entropy in the surrounding area. And therefore, every creation of life generates an increased entropy in the wider system and hastens the progress of the universe towards its final destination. So life is a thermodynamic necessity. We can expect it to occur anywhere in the universe the moment that the conditions are right. Now, again, we need to keep in mind that neither life nor a virus began in a single molecule, a damp little pond getting zapped by lightning or being spontaneously transformed into some miraculous new thing. Chemistry involves concentrations of molecules. So the path from geochemistry to biochemistry must involve reasonable quantities of concentrated solutions, sustained reactions, not limited in location, cascading in complexity over extended periods of time, and in accordance with the normal rules of thermodynamics. Now, if this is going on in this cauldron of complex carbon-based chemistry that have eventually produced cellular life, it's ridiculous to think that vira and viroids and prions produce the same identical chemicals, but in isolation. They all must have come out of the same cooking pot. So there's that common origin. Life is a localized creation of order at the expense of increased entropy in the wider system. Okay, what was LUCA? Well, we've just been looking at that quite a bit. And I want to propose that we should dispense with this once and for all. That's the picture that we've got, and it's wrong. There was no LUCA. Now, Classification began with Linnaeus, and back in 1735, he had 3 categories: plants, animals, and minerals. And under each of those, they were divided into increasingly finer divisions, a sort of tree growing downwards. There's Darwin's initial little sketch in his notebook when he first began to think of his modification by descent. Again, a dendritic branching. And when that was sort of transferred into trees of life, people followed the same pattern. A single root in the ground, dendritic branching, and it first got divided into plants and animals, and then it became more complex as people tried to take into account fungi and microbes. But always that same sort of idea of a tree with one trunk in the ground. Okay, then Coacervates, and we've already looked at this, he came up with the idea of archaea being more closely identical to humans than it was to bacteria. So we got this exciting little system introduced by Carl and George Fox. And then with Fouad Doolittle in 1999, we got this concept of a reticulated network because he identified that there was this business, which already been mentioned today several times, of horizontal gene transfer. And he saw it as a non-linear reaction, rampant, unconstrained, promiscuous horizontal gene transfer. So the trunk of the tree was a network and not a tree. But then, well, got over that, but just going back to that one for a moment. If we progress on from there, we find that as you go further up the tree, again, you have this interactions, and somewhere or over there, one of my diagrams has disappeared, but you get, not only do you get the symbiosis where cyanobacteria and where you get the let me rephrase, you get the cell nucleus and you get the plastid in plants introduced by symbiosis, but then you get continued intermixing as well. Now the same is true of virus. You're going to get that interaction, not just at the basis, but at the ongoing process of evolution. Let's look at this last thing though, and think about what drives evolution. That was the graph I was looking for earlier. You've got down the bottom, you've got the interaction of the network, and out of that you get the beginnings of eukaryotes, and you get plastids and plants. But it's ridiculous to think that those were the only symbiotic relationships they continue. If you look at the human cell or the human body, 8% of our genome apparently is viral. It's being introduced to the humans through viral infections. And so you have a continuing process. Dennis Noble, in his book, The Dance of Life, has a suggestion that horizontal gene transfer has continued. I think he overestimates it and overemphasizes this, but it's a continuing process, and the whole mixture of what we are is much more convoluted than our traditional trees demonstrate. That's why I think we need to take account of virus as a separate tree growing up and interacting with the rest of us. But that brings me to this last point, just what drives evolution. Symbiosis was responsible for the nucleus and eukaryotic shells and for the plastids in plants. So the driving force is no longer conflict, the survival of the fittest, but a sense of cooperation, survival of the fittest community. That applies even to ourselves as humans. Our cells have got organelles. There's a symbiosis there. Our body has organs. Each organ is a symbiosis. The collection of our organs in our body, again, you have symbiosis. Again, on top of that, we require bacteria living in our bodies. What is evolving is not an individual that can survive, but a community and the survival of the fittest community. Now, my proposal, if we acknowledge Vera as a fourth domain of life, it will help us to recognize this symbiotic relationship with the other domains and gives a new look at this whole process. And I think that view has to be revisited and renewed and looked at in a new way. Thank you.
All right. Thank you very much, Bryan. So we have time for questions. And so one question I have is about more on this role of viruses. And so do you think that they could have evolved much earlier on, way before the LUCA, or did they have to come afterwards?
Well, I don't think there was a LUCA. I think if you talk at Doolittle's reticulated network, you have there a network that evolves and eventually slowly develops into 3 separate trees of archaeobacteria and eukarya. And out of that also you have the Vera coming up. Again, not necessarily as one trunk, but something coming up and then diversifying and interacting. It doesn't seem possible to have that complex carbon chemistry going on there, and then the Vera coming out quite separately later on. It's much more sensible that all that came out of the same cooking pot.
Yeah, right. Okay. So I think there's another question. So Eva says, I really like this concept of evolution, cooperation, and survival of the fittest communities. Changes the worldview of evolution and human interactions.
Yes, it's intended to.
Yeah, and she says still puzzled about how to integrate the virus, though.
Well, I am too, but that's why I think, you see, once we got to genomics, when we're looking at bacteria, it's very hard to get any evolutionary sense out of that because they're so similar, as was being pointed out earlier. But once we got into genetics, then it's possible to identify and clarify the different strains of bacteria and archaea in considerable detail. I think if we start applying the same techniques to the Vera, we're going to be able to do the same sort of thing and track how they went and track how they integrated. And I think it's important that we start to do that.
Interesting. There's another question in the chat here. One second. Akanksha asks, if we acknowledge Vera as a fourth domain, where would prions be placed in an evolutionary sense?
Well, they'll be part of that domain. My sense of a Vera as a domain includes the virus, the viroids, and the prions. They are, again, they're very similar in their basic chemistry, although they're different. But it's silly to not group them in one domain and then work out how they interacted with each other and how they interact with the other major domains that we already acknowledge. And again, that's the whole question. I think this is a thing that we need to take more notice of and start looking at in more detail. We've ignored this stuff because we've thought it's not really living, so it doesn't matter. And I think it is living, and I think it does matter.
Right. So there's a few more questions in the chat, and so please, Bryan, feel free to answer them there. I think in the interest of time, we need to move on to the next rapid fire session. So thanks again, Bryan, for that wonderful talk.
Thank you.
All right. So now we're moving on to everybody's favorite part of the day, the rapid fire session. And so we have 5 talks. And remember, you have a maximum 5 minutes for each person. And we got to really keep on time because the total session time now is only 30 minutes. So we kind of have to keep things moving along. And in order to do that, if you once you hit 5 minutes, you're going to hear the Star Trek red alert sound. It sounds like this. I think everybody knows that sound. If you get to 6 minutes, well, just please don't go over 6 minutes. We might have to do something medieval. Okay, so first up, we have Cristian Arenas, I think I said that right, from the Pontifical Catholic University of Chile. So, Cristian, if you're there,
I'm here.
please go ahead and share your screen and take it away.
Okay. So good morning to everyone. I'm Cristian. I'm a freelance biologist, actually. And the purpose of my research is to reflect the incorporation of the theory of autopoiesis and the current strategies to identify the phenomenon of life in the universe in its broad spectrum of how it could be. So,
Hi, Cristian.
Yes?
Hi, sorry, it's just Anna speaking. You're showing us the presenter view, not the full slide view.
I'm sorry. Now?
Nope, still the other way around.
Wait a minute. Sorry.
Might be because you're on two screens, I'm not sure.
I don't know what happened.
Yeah, I don't know if pressing the fourth icon will help you switch between the two.
If
anyone else is more experienced with PowerPoint, please. Yeah.
Now?
Sorry, this is Annaliese. If you go to Advanced, there should be an option to share a portion of your screen. You can just select the part in presenter view that you want to share if you're worried about looking at notes.
Now?
Yeah, this is perfect. Yes, perfect.
Oh, okay. Sorry. So, the current life detection strategies aim to the identification of biosignatures or biomarks. Biosignatures, as we know, is an object, substance, or pattern whose origin specifically requires a biological agent. For example, a pain, a lipid molecule, a microfossil, a stromatolite that makes sense when you search microbial activity, the reflectance of photosynthesis, for example, cyanobacterial, a plant, a tree, a diatomine, express chlorophyll, and the reflectance of chlorophyll is a direct signature of this metabolism. Even the technosignatures speak about intelligent civilization. All these signatures could be grouped in two clusters. First, patterns related to composition, where the key are the components, and the second, pattern related with process. However, I think that exists a third pattern called autopoietic pattern. But what is the autopoiesis? It's a theory about the self-organization of living that offers the advantage of a categorical distinction between the living and the non-living. Since its thesis directly aims to capture the essential organization of life as a constitutive pattern of individual unity, emphasizing the iterative relations of self-production of the component and not in the nature or the structure of the same. This theory was developed in 1973 by two Chilean biologists, Humberto Maturana and Francisco Varela. And summary, the idea is the organisms fit to build themselves, in simple words, and therefore have a unique structural organization that can be identified. How to apply this theory? My idea is take the remote sensing plus machine or deep learning technologies and use it for the detection of autopoietic patterns. This idea is not exclusive. Contrary, is complementary to the current strategy. Taking pattern of composition, pattern of process, and the new pattern of organization, specifically autopoietic pattern, for improve the probabilities to detect germ of life. Thank you for your attention.
All right, thank you very much. You managed to finish on time. I was going to give you a couple extra minutes, but so anyway, we should go ahead and move on. Next up is Helen Greenwood Hansma from the University of California, Santa Barbara. So Cristian, if you stop sharing your screen, I think that will allow Helen to share hers. Yeah.
Okay.
Thank you. Thanks again.
Thank you.
All right. Yep, all good, Helen. Take it away. Oh, Helen, I think you may be muted.
Okay, well, it looks like I'm not muted now.
Okay, now we can hear you. Okay, cool.
Yes, yes. All right.
Wonderful. Okay, please take it away.
All right. So here's a possibility for the origin of life. Did biology emerge from biotite in micaceous clay? This next slide. Where's the next slide coming? Screen share. I'm supposed to.
Okay, now we can see your actual PowerPoint editing mode.
Exactly. Well, I will. Okay. Oh, oops, from current slide. Okay. Somewhere there was a habitat hospitable for everything needed for the origins of life. And this is showing biotite mica sheets with a variety of niches for biological molecules and lipid vesicles. 50 nanometers is 50 biotite or mica sheets. Okay, good. That's, well, at any rate, in this habitat, there were places where metabolic cycles were evolving and places where replication was evolving. Maybe this habitat was between the sheets of the black mica biotite. Okay, and here are lots of little arrows at possible habitats for metabolism and replication with different sorts of molecules, molecular aggregates, vesicles. Well, and then the vesicles, as it says, are membranes. So here on a larger scale, 5 microns, what's that? Thousands of mica sheets. Membranes were forming and encapsulating molecules that accumulated between the biotite sheets. After a long time, occasionally a membrane would encapsulate everything needed for a living cell. So maybe one of these is a living cell. So what's biotite? Biotite is a mica, which is a clay mineral. Potassium bridges the sheets. The sheets are negatively charged. You can see 2 brown. Okay, there's 2 brown biotite sheets. There's little mica sheets, biotite sheets, more biotite sheets, potassium ions. And biotite has a lot of iron, which is good for redox reactions. It's its main advantage for the origin of life over mica or other clays. So the only important thing in this slide is that here, this is an 80 micron drawing of this. And here is this 80 micron picture. And 80 microns is this tiny, barely visible mica fragment from micaceous clay. So here's micaceous clay in the middle of a wet dry cycle. And here's mica that was washed out of the micaceous clay. So I have a lot of similarities that I find between life and mica. I talked about the potassium ions that are in high concentrations in all our cells. This one's huge. This is work by Hassenkam this year with Damer. You put nucleotides in an aqueous solution on a mica surface, run it through wet dry cycles, and they polymerize into RNA. And obviously we know we need some way of forming RNA and DNA in order to get life. So the mica surface has a negatively charged crystal lattice with a spacing of a 0.5 nanometer. And the extended single-stranded nucleic acids have also a negative charge with a spacing of a 0.5 nanometer in the phosphate groups. It makes sense that there was a mineral surface that templated all these biopolymers. And maybe that 0.5 nanometer grid is why we have 3' 5' DNA and not 2' 5' and not pyrophosphate in order. And okay, so that covers it. There's lots of other fine print here, but that's, oh, okay, one final point. There's not a lot of micaceous clay on Earth at that time, but there doesn't need to be a lot. You can think, for example, of borate that Steve Benner has been talking about, and Bob Hazen finally agrees that, well, there might have been enough borate even if there wasn't much. So I know there's not a lot of micaceous clay at that stage, but that doesn't mean there was none or that a lot was needed for the origin of life. Thank you.
All right, perfect. Thank you very much, Helen. Finished right on time. So next up, we have Gabriel Gonçalves. I don't know if I said that right.
I'm here.
And please take it away.
Okay, let me share. Is it showing?
Yep, all good on my end.
Okay, let's start. So, good morning, everyone. Thank you very much. My name is Gabriel Silva. I'm going to present a work called UVC Photoprotection by Ferric Ions, Consequences on the Habitability of Ancient Mart Ponds. This work was developed at the University of São Paulo, Brazil, and is a part of a bigger project about the habitability of Mars. Well, areas on the surface of Mars show evidences of possible aquatic acidic environments rich in soluble ferric ions in the past. On Earth, such environments can bear organisms like the extremely acidophilic iron-oxidizing bacterium, Acidobacillus fluoxidans, a very sensitive organism to UVC. It is calculated that the maximum flux of UVC into 254 nanometer band in Mars would be more or less 22.6 milliwatts per square meter. However, soluble ferric ions have a high absorptivity in the UVC range. This work evaluates the effect of ferric attenuation of UVC on the protection of Acidobacillus fluoxidans for modeling of Mars ponds with different ferric concentrations and prediction of habitable depths. I'm going to explain it more later. The methodology we use to do this, we use the Acidobacillus fluoxidans in a carbon-free minimal medium with iron, with different concentrations of iron to grow. So we expose these cultures to different doses of UVC into 254 nanometers band. And to quantify the concentration of cells that survive it, we use the most probable number method. So first of all, we use the Beer-Lambert law to calculate the molar absorptivity of ferric ions. And we got on the UVC 254 nanometer band. And we got something more or less 2.4 liter per centimeter millimole. Also using the Beer-Lambert law, we get an equation that correlates the median lethal dose for survival of this microorganism with and without iron, the molar absorptivity and the concentration of the ferric ion, and the optical path length of these solutions. So we calculated the survival curves for 4 concentrations of iron, and we got the median lethal dose for this microorganism in these 4 concentrations. With this and the equation, we calculated the lethal dose without any iron for Acidobacillus fluoxidans for UVC. It was 17.6 joules per square meter. So using these results and this equation, we try to calculate the habitable depth of the spawns on Mars. We call a habitable depth the point where the time necessary for a medium lethal dose with that iron concentration be equal to the time necessary for the microorganism to duplicate. So the growth rate and the death rate was the same. And from that point on, the growth would be higher than the death by UVC. Using literature data from Carvalho and colleagues, we found that he studied many pounds on Earth, and the minimum concentration of iron was 0.03 millimoles and maximum 18.9 millimoles. And we used previous experiments of growth rate that got in our laboratory of the maximum growth rate was 0.19 per hour. We also simulated half of that growth rate and 1/10 of that growth rate. The results varied a lot. So for the lowest growth rate with the minimum iron concentration, we need more than 10 meters to protect the cells from the UVC. However, with the maximum concentration of iron and the maximum growth rate, we just need 1.6 millimeters of liquid above the cells to provide the protection to UVC. The maximum concentration of iron is not that much. And if you use that concentration and the lowest growth we propose, we still have just the 1.6 centimeters of liquid to protect the cells. So this work shows that soluble ferric ions can provide great protection to UVC for sensitive organisms like Acetobacillus fluoxidans. In a future work, we want to use more complex simulations and experiments using UVC, UVB, and UVA during a more complete simulation of the Martian day cycles. That is it. Thank you very much.
All right, thank you. Perfect timing. And yeah, if you have any questions, please feel free to go to the chat or Slack. So thank you again. Next up, we have Adrian Barry-Sosa. So please go ahead and share your screen. And yep, I can see it. And
all right, perfect.
Yep, all good. Take it away.
Hello, everybody. Today I'm going to talk about the main topic of my PhD research, which focuses on the Upper Floridan Aquifer, or UFA for short. The UFA is a karstic aquifer that extends throughout the whole peninsula of Florida, as well as some part of southern Georgia and Alabama. In modes of extension, it's a confined aquifer, meaning that on top of the rock layer that contains water, there is another rock layer that is impermeable, that doesn't allow water to go through. However, in north central Florida, that top confining unit has been removed by erosion. And there, water from the UFA is discharged to the surface throughout many springs. From all those springs, we have picked up 5 that are representative from the geochemistry that it's found at several points in the UFA. What is the geochemistry about? Well, UFA water has a pH of around 7. In all cases, is undersaturated in dissolved oxygen, although the actual concentration varies significantly between different springs, from 3% all the way to 50% dissolved oxygen. Residency times in those places where it has been estimated, it's around 30 to 40 years. And the dissolved oxygen concentration, dissolved organic carbon, sorry, it's very low. It's in the range of oligotrophic conditions. And those oligotrophic conditions are translated as well to a low microbial cell density for 10 to the 6 to 10 to the 7 cells per liter. I, of course, don't have a lot of time to go into the details of the microbiology of the UFA, but I just want to give you a couple of highlights. First of all, when we measure the diversity of the microbial communities using the Shannon index, we find that the correlation with dissolved organic carbon is negative. And this is a striking result because not very environments, in fact, not surface environments are known to have this correlation, although it has been observed in other subsurface environments as well. We don't know yet why this is the case, but we suspect that this could be a response of microbial communities to cope with the oligotrophic conditions they live in. The second main highlight that I want to bring on the table about the UFA is the presence of two groups, the group Dipan that belongs to the domain Archaea and the group Pathecibacteria that belongs to the domain Bacteria. And in spite of belonging to two different domains, these two groups share common characteristics. One of those is that they are small. They are small enough that in some cases, they are able to go through a 0.2 micrometer pore size filters and be retaining in the 0.1 micrometer fraction. In some of these 0.1 micrometer samples, they can make up to 70% of the population. So they are fairly abundant in some of the springs we have sampled. But why is the UFA relevant from an astrobiological point of view? Well, in this subsurface environment, there are many geochemical processes that are triggered by microbiology that can be used as biosignatures. It has been observed that nitrous oxide is above atmospheric concentrations in all the springs that we have sampled. And we strongly suspect that nitrous oxide production is linked with microbial activity. So it could be a potential gas to be used as a biosignature. We also suspect that methane could be produced here, although we don't have a strong proof of that yet. Another potential biosignature is a mineral goethite, which is precipitation is also suspected to be related with microbial activity. And the second main point why the UFA is relevant from an astrobiological point of view is that it can help to understand adaptations of life to cope with these oligotrophic conditions. Oligotrophic conditions that are likely to be present in similar environments, similar underground environments, like underneath the ice shelves of Europe and Enceladus, as well as underneath the ice cap of the Martian South Pole and some caves on Mars. And with all of that, I would like to thank every one of you for your attention and acknowledge all the people that has helped me in this research. Thank you very much.
All right, beautiful talk, Adrian. Thank you very much. And so last but not least, we have Annaliese Meyer. So if you can share your screen.
Oh boy.
All right.
Okay,
Very
awesome.
good. Looks good. Take it away.
Tēnā koutou. I'm Annaliese Meyer, an incoming PhD student at the University of New South Wales, speaking to you today from Aotearoa, New Zealand. I'm going to discuss the reality of planetary protection measures in the current era of private space startups. This report is the result of ongoing research from the SGC 2019 Space Exploration Working Group. So the current space sector has a huge range of privately held businesses. In this slide, the icons are scaled to the total funding of the business, with Blue Origin coming in at $5.5 billion. Now, when I'm discussing private businesses, I'm explicitly referring to those without an IPO or initial public offering. This means they're not available for public investment on the stock market and thus are almost entirely funded by venture capitalists, government contracts, and importantly for the largest two companies, by so-called angel and super angel investors. Today, I'm going to focus primarily on how early stage startups, so the tiny little dots in the corner here, interact with existing planetary protection regulations and what needs to be rethought in order to encourage innovation in the deep space sector, prevent irreparable harmful contamination, and ensure accountability on the part of both large and small space businesses. So if we zoom in on one of the smaller companies here as an example, I'm going to talk about Moon Express briefly. So it's probable that even more initially small satellite companies will venture into the interplanetary sector, as we've seen with Rocket Lab and iSpace. When we look at the current record of funding for Moon Express, it comes in at about $51 million. For comparison, the Viking lander Gold Standard planetary protection measures are estimated to have cost about $100 million. Well, in the US, NASA might be able to provide some support, the complicated dynamics of public-private partnership are likely to become even more muddied as private interplanetary missions develop. So that's the stage. Planetary protection is expensive and is often an overlooked cost in developing startups, and I'm sure I don't have to convince all of you that it's vitally important to the future of astrobiology. So given the rapid nature of this talk, I'm not going to go through all our findings and recommendations at present. Instead, I'll focus on a particular case study and highlight some of the recommendations that followed from that instance. So one of the major findings from this group is a bit of a double-edged sword. Private space startups are the leaders in making access to low Earth orbit and eventually deep space more affordable and accessible, both for research and commercial purposes. This is done through a combination of innovative techniques and pure scrappy fighting spirit. So the case study I'll chat about is the use of carbon fiber composites for building entire launch vehicles and capsules, born out of direct interviews with several early and mid-stage launch providers. Several small-sat launch providers have begun using largely composite rockets to great success, such success that some of these, like New Zealand's own Rocket Lab, are now actively planning interplanetary missions. While on an engineering level, these innovative techniques will likely scale up to interplanetary missions well, such as even been explored for the Orion capsule, they have not been sufficiently investigated for their compatibility with microbial reduction techniques. The need to protect intellectual property prevents independent researchers from performing studies on the particular surface properties of the finished materials. Rigorous characterization of resin properties and even the microscale roughness of the surface can have important implications on the efficacy of cleaning measures, such that investigation of composites in publicly funded mission segments, such as the Blades of Ingenuity, for example, are insufficient. So to expand on this, liquids follow different wetting regimes based on surface microstructures as shown here, which changes the liquid penetration and contact angle of the cleaning agent. Certain surface textures or finishes might provide unaccounted for refuge for spores and organic burden. Our group is looking to illustrate this through a range of composite finishing and impregnation types with a series of simulations similar to this one constructed via lamps. Further, bacterial adhesion might be affected, as shown with microscale patterning, causing different adhesion levels in previous works. So characterizing this fully is a pretty considerable task and requires use of electron and/or atomic force microscopy, molecular dynamic simulators, and further microbiological assays. Small startups generally don't have the capital or the expertise required for these sorts of studies, and as mentioned before, IP protection of proprietary manufacturing and QA techniques prevents public research labs from taking on these studies. Commercial interplanetary missions are likely a reality of the near future. So do we restrict access to those companies with sufficient backing from single super angel investors to perform these sorts of characterizations in-house? Do we sacrifice potentially groundbreaking science by loosening planetary protection measures to stimulate economic development, as has been proposed by multiple private stakeholders, or is there another option? So this brings us to the recommendation that came out of this case study. We propose that public funding or fiscal incentives such as tax breaks for private investors be provided to create national centralized facilities to be contracted for company-specific planetary protection research. This could include clean room space and necessary microscopy facilities. These would be managed by an external source and specifically contracted for missions. Facilities could run comparably to shared lab space or startup incubators with resident technicians to perform necessary analyses and prepare internal protected reports. We do observe that investing in shared planetary protection facilities might stimulate interest in innovation in interplanetary missions among private companies, much in the same way that business accelerators and sandboxes promote rapid growth of early stage startups. This investment would be particularly impactful in nations with a small but growing private space sector, such as Canada, Australia, and India, among others. So that brings me to the end. If you're interested in finding other findings and recommendations from this group, please do contact me or watch for our talk at Coacervates 2021. And thanks to all of my collaborators and the Van Kranenot, Wang, and Burns lab, as well as all the groups at U of A that have been supporting me through this.
All right, brilliant talk. Thank you very much, Annaliese. So that pretty much wraps up our rapid fire session and the first session of today. So I just want to thank all the speakers again. Y'all did a wonderful job. So there's a round of applause for y'all. And yeah, so we're going to meet again here at 12:30. So please come back.